Good morning. Welcome to IBG, and it's my pleasure to introduce you today, our guest speaker, Stefan Pak'ı. And as you know, in IBG, as a multidisciplinary research excellence center in Izmir, we are running a EU-supported rare disease project, RareBoost, and to develop the research and soft skill capacity building and try to solve unsolved and undiagnosed rare diseases. And through this, we are collaborating different organizations, and one of it is Undiagnosed Disease Network International. And we found the opportunity to meet with Dr. Pak'ı many years ago, let's say, and in our program we are collaborating with him for the functional in vivo studies because Stefan is working in. Actually, he's a professor of pediatrics and co-director of Undiagnosed Disease Network, UDN, elegans model organism screening center. And he has earned his bachelor in Sydney and then worked in Australia. And then prior to his appointment now in Washington University, he held a faculty position in Harvard and also Pittsburgh. So I asked him to introduce also a little bit about UDN and how it's working. And thank you for his collaboration and coming and visiting us. Please.
Okay, thank you for that introduction, Dr. Özbek. Before I start, I'd like to say goodnight and also thank you to Dr. Özbek and his team for providing me with this wonderful opportunity to visit the beautiful city of Izmir and IBG. And I've heard a lot of great things about IBG already, and I'm looking forward to learning more about the center as I meet with some of you a little later on. So this morning, I'd like to speak to you, and I'm very excited to share with you work we're doing at the Washington University in St. Louis. We have a center there called Center for Rare, Undiagnosed, and Genetic Disorders. And also, I'd like to share with you work we're doing at the Undiagnosed Diseases Network, which I'll be referring to as UDN. Some of you may have heard about the UDN, but for those of you who may not be familiar with it, I thought I'll provide a very brief introduction. So the UDN is a large a research collaboration network in the United States. It first started in 2008 at the NIH as an Undiagnosed Diseases program led by Dr. Bill Gall. It then established into a Undiagnosed Diseases Network in 2014, into a multi-center network, which consisted of 7 clinical sites, 2 model organism screening center, a central biorepository, and a coordinating center, as well as a metabolomics center. And over the years, since then, it has multiplied to now to be 24 clinical sites scattered throughout the United States, 3 model organism screening centers, and 6 data management and coordinating centers. And so the names of some of the centers are shown here. And so the UDN utilizes standard evaluation protocols, and this is to ensure uniformity across all the clinical sites. So they evaluate the patients and all the procedures are the same so that we can disseminate the information with each other and everything stays uniform. The UDN promotes extensive data sharing and expert collaborations, shown here in team science. And it incorporates advanced genomic strategies and technologies, including functional studies in model organisms. And I'll talk more about these later on. And the whole purpose of the UDN is to solve medical mysteries and to bring diagnosis to people living with rare undiagnosed disease. And so this is the UDN workflow. So individuals that are suspected to have a genetic disorder can apply free of charge to the UDN coordinating center. And then they are evaluated in terms of the form that they fill out. And if it is determined that they are likely to have a genetic disorder, they are then referred to one of the clinical sites, depending on the geography and the location. These individuals are then receive a comprehensive clinical evaluation, which includes a deep clinical phenotyping, a detailed analysis of the family history, and trio genome or exon sequencing, and sometimes a multi-omics analysis to see what is going on in the patient. And in about 25% of the cases, these individuals receive a diagnosis at this point in time. And so this is the facts and figures for the UDN so far, and it's accurate as of April 2026. And so far, more than 8,700 cases have been received, and of those, 3,600 individuals cases have been evaluated. And about 2,500 individuals have received exome or genome sequencing, and 534 of them, of the participants, have been evaluated by the Model Organism Screening Center. And in total so far, over 1,000 individuals have received a positive diagnosis. And we have, as a whole, published 335 manuscripts. So I mentioned earlier that around 25% of the cases are actually solved by the clinical sites following the evaluation and analysis of the genome. However, there's about 70 to 75% of the cases that remain unsolved. And in some of the cases, those cases are referred to the Model Organism Screening Center for further evaluation. And so the Model Organism Screening Centers that are part of the UDN are, we use 3 model systems, C. elegans, and this center is at my university, at Washington University in St. Louis. We also have a fruit fly core, which is at the Baylor College of Medicine. And then we also have 2 zebrafish cores, again, one at Washington University in St. Louis, and another one at the University of Washington. So it's confusing, but because Washington is the first, president of the United States, so a lot of universities and towns and states are named after Washington. But we're separate institutions. Okay, so this is the Model Organism Screening Center workflow, and I don't want to bore you. There's a lot of information there, but we actually evaluate approximately 200 variants per year. There's an initial review by Baylor and WashU bioinformaticians based on the medical genetics to see whether it's suitable for model organism studies. And then there's also in parallel core level analysis that's performed by the worm, fly, and the fish cores. And the model organism working group meets once a month to discuss these cases to see which one of these model organisms will model the case. And a organism is then recommended to study the variant, and clinicians are then informed. And then the model organism researchers will meet with the clinicians to discuss how we will model those cases. And if there is agreement, the core will then be committed to doing experimental studies. Okay. So as shown here, it usually takes between 5 to 12 months to perform these functional studies in the model organisms. And worms usually take 5 to 6 months, flies 6 months, and fish 12 months. These values are in theory, but in practice, we perform lots of additional studies, including trying to understand the genetic mechanism, and also to do enough studies to get a publication, which is important for the researchers. And so that usually takes additional time to get the manuscript ready. So that was just a very brief introduction to the UDN. And with that, I'd like to shift gears a little bit now and talk to you about how we use C. elegans, which is my favorite model organism, to perform functional studies and on the candidate disease genes. Okay. So this is the animal that I work with. It's called Rhabditis elegans, and it's so named because of its elegant movement as it crawls across the agar plate. It's a microscopic nematode that lives in the soil. And just to get an indication of the size here, I put the worm here, a little dot next to the Turkish kuruş, just so that you know the relative size of C. elegans. It's very tiny. And it's attracted to sugars and microbes. And so in the laboratory, we grow these animals on a Petri dish with agar, with a little drop of E. coli. That's all you need. And on Friday afternoons, before I go home, I'll transfer 3 worms onto the plate. And by Monday, I have thousands of worms crawling around. So it's very rapid. It grows very quickly. And that's why it's been used as a genetic model system for many decades. Oh, I forgot to mention that that I know Turkish people are very fond of figs, right? Well, so are C. elegans. So you may have encountered them or maybe even ingested them without you knowing, because some varieties of Rhabditis are attracted to figs. So wash your figs carefully before you eat them. But it's okay if you eat them because they're not parasitic in any way. Okay, so then even though worms and humans are very different, many of you might be asking, what is the utility of using C. elegans in rare disease genetic research? Okay, believe it or not, worms and humans are actually very similar at the genetic level. Humans have 20,000 protein coding genes. Worms also have 20,000 genes, and there's about 50% homology between worms and humans. And so you have 1 in 2 chance that you'll find the human gene in the worm. Okay, and for disease-causing genes, that percentage is a little higher, sometimes like 60 to 70%. Okay, and so that's why worms have been used as a genetic model for many decades and to model human diseases. In addition, many of the fundamental biological processes are well conserved in C. elegans. For example, genes that control programmed cell death or apoptosis were first identified in C. elegans. And also, as a result, many genes and pathways have been studied in C. elegans. And in particular, new genes that are involved in the endocytic pathway were discovered in C. elegans. And even insulin signaling pathways and aging have been heavily studied in this model. In addition to the genetic similarities, worms are actually very easy to manipulate genetically. So we can generate CRISPR knock-ins and knockouts in a matter of days, as opposed to months and years in higher model organisms. They have a very rapid generation time, as I mentioned earlier. And so we can obtain knockouts, as I said, in a very short period of time, and even outcross them twice within 3 weeks. Also, worms in the worm community, there's a lot of reagents available, and null deletions exist for many of the C. elegans genes already. And then we can just purchase them from the C. elegans stock center for $7. And if the deletion doesn't exist, we can ask the center, the CRISPR knockout center, to generate them free of charge. Okay, so there's a lot of resources out there. And also the null or loss of function phenotype for about 10,000 genes are already known. So there's a lot of information out there already to tell us about the gene function. Okay, this is the schematic showing how we model candidate gene variants in C. elegans. So if an undiagnosed patient undergoes trio genome sequencing, a candidate disease gene may be identified and a variant associated with this gene can be nominated. And however, these are, even though the bioinformatic tools have improved, these are still predictions. And so we're not 100% sure where, especially if this gene is a gene of uncertain significance or a gene that has never been associated with a disease before, okay? So there's always a question mark there. So in order to answer this question, we model in C. elegans, and this is how we do it. We identify the ortholog in C. elegans. And as I said, you have 1 in 2 chance of finding an ortholog. And then we use CRISPR-Cas9 to knock in the human variant into the worm gene. And following that, we then perform functional studies or phenotypic analysis to determine if there's anything wrong with the worms. Remember, using the CRISPR-Cas9 approach, we're changing 1 base pair out of 100 million total base pairs in C. elegans. And so if there is a phenotype in the worm, then there's a very, very good chance it's caused by this variant, okay? And so that's our approach. And so the information that we gather from the phenotypic studies can lead to a molecular diagnosis in the patient. But some of you sitting there might be wondering, worms and humans are so different. You know, how can you use worms to gain any useful information about the human disease? And I would agree that worms are very different. Worm phenotypes are very simple. You have short worms and long worms. You have worms that move slowly. You have worms that are uncoordinated, that have problems laying eggs. But you know, there aren't the complex phenotypes that we see in humans, right? Like microcephaly, intellectual disability, hearing loss, dysmorphic features. And so the phenotypes that you see in worms do not resemble the phenotypes that you see in humans. And so often, this is the reason why a lot of clinicians don't want to use C. elegans as a model system. But I would argue that worms are still extremely useful in understanding a disease gene. And here's an example here. Even though worms are invertebrates and they don't have bones, okay, we can actually study a craniofacial disorder in worms, okay? So my question was going to be, what's common between a human craniofacial disorder and a worm that has an egg-laying defect phenotype, okay? And oops. What's common is that these phenotypes are caused by the same gene. So in humans, mutation in this gene called TWIST1 causes a disease called Sweeney-Cox syndrome, characterized by craniostenosis. In worms, mutations in the same gene called HLH8 cause an egg-laying defective phenotype. And so these two phenotypes don't resemble each other, right? However, they're called phenologs because these are phenotypes that are very different but arise from genetic disruption of the orthologous genes, okay? And these phenologs are extremely important because, first of all, they tell us that this mutation in this gene is damaging, right? Because the worm's not normal. So immediately you know that the variant is damaging. But second of all, we can exploit the advantages of the C. elegans model to perform genetic screens, suppressor or enhancer screens, to identify other players in the pathway, okay, other genes that are targeted by this twist1 homolog. And what's interesting is that in humans, once they catch up, the downstream targets of twist1 were also found to be FGF receptor, Notch ligand, and TBX transcription factor, which were identical. So the phenotypes are very different, but the molecular process or the genes that control those phenotypes are the same, okay? So I want you to keep that in the back of your mind. Okay, so the goals of the C. elegans studies, therefore, are not to say that this gene variant is definitely causing the phenotype in the patient, like, for example, microcephaly or intellectual disability, because we can't model that in C. elegans. However, we can provide experimental data in support for the candidate gene variant being damaging to the gene's function, okay, which is very important. Secondly, we can determine the genetic mechanism of the variant. So is it a haploinsufficiency? Is it a loss of function phenotype that results in haploinsufficiency? Or is it some sort of change of function in the gene that results in a dominant negative or a gain of function phenotype? And then finally, we try to provide some initial insight into the molecular and cellular mechanism of the gene variant. Okay, so these are the three deliverables that the C. elegans core promises to the clinicians. So now I want to shift gears again and talk about an actual unsolved case that we were able to solve using the C. elegans model to sort of highlight how worms can be useful for this process. Okay, so this was a collaboration between Washington University and Baylor College of Medicine. So this was the clinical site, and we were the model organism screening center. The clinicians are shown here, and the patient was a 15-month-old female with global developmental delay, dysmorphic features, clubbing of the digits and toes, and interstitial lung disease, reminiscent of a surfactant disorder. Unfortunately, as we were working on this case, the patient passed away shortly after. However, we continued working on this case because we felt it was very important to find a diagnosis for the family. So initial clinical evaluation of the genome came back negative. However, a research reanalysis of the genome then identified this de novo heterozygous variant in this gene called RAB5B. RAB5B had not previously been associated with a human disease, and the impact of this variant was unknown to the gene's function. So I want to mention that the clinician, the clinician who saw this patient described that the patient's lung samples had this aberrant surfactant B staining, which was reminiscent of the hereditary surfactant B mutations. Okay, so surfactants, as I'll mention later, are important for lung function, and mutations in these genes can lead to fatal neonatal respiratory failure. However, what was interesting was that in this case, even though it looked like a typical surfactant disorder, no genes that are known to be associated with disorder were identified in this patient. So this case was then passed on to the C. elegans Model Organism Screening Center to perform functional studies. So as I mentioned, the patient has lung disease, and this RAB5B gene was identified, and there was a variant called D135H, and its functional consequences were unknown. So then we modeled this variant in C. elegans. Luckily, the RAB5 gene is present in worm, and it's highly conserved. As you can see, the multiple sequence alignment between C. elegans and human. And this D here, aspartic acid, is the residue that was affected in the patient, and that's also conserved. And it happens to be in this green region which is the nucleotide binding site of this protein. And so we felt that this mutation would likely to have a bad impact on the protein function. So we use CRISPR-Cas9 to introduce this mutation into the C. elegans Rab5 gene. And then we perform functional studies to determine if there were any abnormalities. We usually generate several strains for study. First of all, we generate 3 independent lines of the patient variant edited animals. We also generate 2 control lines, which are essentially just controls for synonymous changes that we introduced. And then also we generate a null mutation so that we can compare it to the patient variant. And we call this the deletion mutant. Okay. And the first thing we do is just look at organismal phenotypes. We look at whether they look normal, whether the body shape and size looks normal, or whether they move through development normally. Do they reproduce correctly, or do they move in a different way? So these are some of the organismal phenotypes that we look at. And again, these phenotypes don't have anything to do with the human phenotype, but they are a good readout for gene's function. Okay, it tells us whether the gene is working correctly or not. And so the first thing we did was look at development in C. elegans that carries this mutation. And normally worms, grow from a fertilized egg to an L1 stage larvae in about, I think, 14 hours or so. And then they undergo 4 molting cycles to become a mature adult. Okay, so in a wild type animal, they grow normally, obviously, and become fertile adults. However, in worms that carry the patient variant, they were extremely abnormal in their development, and they died as early larval stage worms. So we knew immediately that this human variant was damaging to Rab5 function. So we knew that within 2 to 3 weeks of starting these studies. Okay, we could have finished there, but we wanted to continue going. So we provided experimental data in support of the candidate gene being damaging. Next, we wanted to understand the genetic mechanism of the variant. And to do this, we analyze some of their quantifiable phenotypes, such as their locomotion. And this is a device that we use called WormLab, and it's a video recording device that records the worm's movement, and it can calculate automatically the size, the speed of that the worms are moving. And so these are and the body bends and whether they have the same wavelength or amplitude. And so it's very, very quantitative phenotyping method. Okay. And so when we did this, we found that, previously we were just looking at homozygous animals to see if the gene function has been damaged, okay? But now we know that the patient is a heterozygous individual, right? So we wanted to know what the heterozygous phenotype was in the worm. And this is important to understand the genetic mechanism. Okay, is the genetic mechanism consistent with the dominant presentation in the patient? And so we observe their locomotion, and you can see in healthy animals, as I showed earlier, they move normally and they move fast. However, in the patient variant, this movie is moving, but the animal is basically staying in the same spot and unable to move forward. Okay, so even as a heterozygous state, this variant was extremely damaging to the animal. And this is our quantification. So these are the 2 CRISPR edits that have the heterozygous patient variants. And you can see that they move at a much slower speed compared to the wild type here in the gray and the control edit here in blue. What's really interesting is in the null heterozygous or the deletion heterozygous, there is no phenotype, they're wild type. Okay, so that means that this gene is not haploinsufficient in C. elegans, but the patient variant has a dominant phenotype, which is not present in the null phenotype. So it's doing something different. So this is when we knew that this variant here that we're working with, because it displays a dominant phenotype not observed in the null or deletion heterozygote, then it must be some sort of a change of function allele, likely a dominant negative. Okay, and this is where worms are very powerful. Now we can go into the genetics and figure out, is this really a dominant negative allele? And the way you test for dominant negative mechanism is that dominant negative phenotypes can be suppressed by addition of extra copies of the wild type allele. And so we can easily do this by, we have the endogenous RAP5 gene on chromosome 1. We can insert another copy of the wild type RAP5 gene in chromosome 2, and then introduce different copy numbers into the worm and see if the phenotype is reversed. And we performed RNA-seq to make sure that the endogenous gene and the extra transgene that we made were being expressed at the same level. And so when we do this study, we notice that when we have 0 copies of the extra copy, the animals move very slowly, as I showed earlier. But when we add 1 extra copy and 2 extra copy, we see full rescue of the phenotype. So in this case, the last column, we need 3 copies of the wild type allele in order to suppress the patient variant. And so by definition, this patient variant must be a strong dominant negative. Okay. So we use the worms to determine the genetic mechanism, and now we want to move on to provide some insight into the molecular and cellular mechanism of the gene variant. As some of you may know, RAP5 is a well-studied protein, and it's involved in endocytosis. And it's important for fusion of these early small endosomes into these large early endosomes. So one of the first things we did was then look at endosomes in C. elegans. And we labeled endosomes using this 2x5 GFP reporter. And in normal cells, you see endosomes that have these small dots, larger circles, and bigger circles, which are indicative of this fusion process where you go from small endosomes to large early endosomes. However, in the patient variant, we knew straight away there was some defect in that process, because now all we see is this accumulation of these little small dots, but not those bigger endosomes. Okay, so we knew that there was some defect in the endosome formation in the patient variant. We then asked, is the whole endocytosis process defective in this worm? And so to do this, we used another marker which secretes GFP from the muscle cells into the body cavity. And then there are these cells called celomocytes that essentially scavenge all these GFP from the body cavity, and so they light up green. However, if the endocytosis process is defective, then we see lots of green accumulating in the body cavity. So this is a normal worm that's expressing this transgene, and as you can see, the celomocyte is green, but the rest of the body is fine. But in the patient variant, we see that there's lots of GFP accumulating in the body cavity, which means that endocytosis cannot happen. And that's why now all this material is accumulating in the body cavity. And so we conclude that this patient variant animals are defective, not only in the endosome formation, but also in the endocytosis process. So we've provided some initial insight into the molecular and cellular mechanism in the gene variant. And then the next question we asked was, well, what is the connection between the RAP5 patient variant and interstitial lung disease? Because that's not a question that we answered so far. To answer this question, we collaborated with 2 doctors at WashU, Steve Brody and Jen Wambach. Steve Brody is a pulmonologist, and Jen is a newborn medicine neonatologist, and she specializes in neonatal respiratory failure and child interstitial lung disease. And if you recall what I said earlier, the clinician that saw the patient said that the patient had aberrant surfactant protein B and C staining reminiscent of a hereditary surfactant protein mutation. However, mutations in genes that are known to cause the interstitial lung disease, particularly through surfactant mutations, were not identified in this program. So what's going on in this program? Some of you may know what surfactant proteins are, but surfactant proteins are critical for lung function. They're expressed by alveolar, type 2 alveolar cells, and then they secrete this surfactant protein and they coat the lungs, and they maintain the surface tension required for proper breathing. And so if you don't have that surfactant there, then the lungs can collapse and the baby can die very early on. Surfactant proteins are produced in the ER, Golgi pathway, as precursors, pro-surfactant B and C, and then they are packaged into multivesicular bodies, and finally into lamellar bodies, where they are processed into mature forms and mixed with phospholipids and then secreted into the lung. As I mentioned earlier, any mutations in these surfactant protein B or the ABC transporters that allow phospholipids into the lamellar body lead to severe interstitial lung disease and sometimes failure in neonates and death. So one of the first questions we asked was, well, is RAB5 said was that the patient has type 2 cell hyperplasia. So you can see that there's hyperplasia in the lung cells compared to here. And then when we looked at RAB5B staining, in the normal control, we see lots of RAB5B, but in the patient, we see a significant reduction in RAB5B shown here. Okay, so for whatever reason, the variant in RAB5B is causing there to be less of that protein in the lungs. We then asked, does RAB5 play a role in surfactant protein maturation? And so here in normal lung cells, you see that the pro form of the surfactant protein B is present, and then the mature form is there as well. So that we can tell that there's correct processing of the surfactant proteins in the normal cells. However, in the proband, there is production of the pro-SPB. However, the mature form is almost missing. And this is the quantification here. So not only does RAB5 decrease mature surfactant proteins, and I'm not showing surfactant C here, but also surfactant protein C also decreased in the patient. And so we hypothesized that RAB5 plays a role in pro-surfactant trafficking. And to prove this, we used a cell culture approach to knock down RAB5B in these cells called MLE15, which are, I believe, mouse lung epithelial cells. Okay, so these are cells that normally produce surfactant proteins, and so we thought it would be a good model to use this to test the role of RAB5. So we used shRNA treatment to knock down RAB5B, as shown here in lane 2 and 3. And when we knock down RAB5B, we also see that the mature SPB, or the surfactant proteins, significantly decrease. Okay. But the pro-SPB proteins remain the same. So from this, we can conclude, that RAB5B plays an important role in surfactant processing. Okay, so in disease, and we identified a variant in RAB5B. We then performed functional studies in C. elegans to determine whether this variant was causal, and we found that RAB5B in C. elegans is damaging and produces a very strong dominant negative product. We also found that RAB5 heterozygous animals were defective in early endosome fusion and endocytosis process. And then when we looked at the human lung samples, we found that RAB5B expression was significantly reduced, and that precursor surfactant processing was severely disrupted in proben lungs, leading to surfactant dysfunction. And this led to the discovery of a new function of RAB5B in the regulator secretion pathway. And this is just an example of how functional studies and diagnosis of rare diseases are very, very time-consuming and challenging, and requires a lot of resource investment and time investment and collaboration and sharing of data. But this also highlights the fact that once we published this N of 1 case, we were then a couple of years later contacted by several other clinical sites. This really importance of collaborations, and I really appreciate the collaboration that we started with Dr. Özbek's laboratory. And we hope that through the UDNI and through RareBoost and other initiatives that we can continue to advance rare disease research one gene at a time. And these are the people who did the work. I want to show that photo. So these are the people in the lab who did all the work. And these are all the people that I'd like to acknowledge. The Baylor clinical side, people in the C. elegans screening center, especially Tim Shadle, who's the co-director of the center along with me, and the other clinical collaborators are shown here. And finally, we could not have done this at WashU without the support of the dean, who matched the funds that the NIH was contributing to this project, and also the chair of Pediatrics, Gary Silverman, who was a strong supporter of the undiagnosed research, as well as the Model Organism Screening Center.